All right, class, let's begin with a fundamental question. What exactly is cancer? At its core, cancer is not just one disease, but rather a collection of diseases characterized by the same underlying feature: uncontrolled cell growth and division. Normally, our cells are like disciplined students in a classroom. They grow, divide, and die according to very strict rules laid down by the body. These rules are enforced by regulatory proteins and checkpoints in the cell cycle, much like a teacher ensures order. Now, in cancer, these rules are broken. A single cell acquires mutations, changes in its DNA, that allow it to bypass these restrictions. Once this happens, that rogue cell can multiply when it shouldn't, ignore signals to stop growing, and even avoid natural cell death. Over time, this small group of abnormal cells can expand into a mass, which we call a tumor. But tumors are not all the same. We classify them into benign and malignant. Benign tumors grow but remain confined to one site. They don't invade surrounding tissue or spread to distant organs. Malignant tumors, on the other hand, are the true cancers. They not only grow uncontrollably, but also have the potential to spread. This spreading is called metastasis, and it's what makes cancer life-threatening. Now, what causes this transformation from a healthy cell to a malignant one? There are multiple causes. First, there are genetic factors. Inherited mutations in certain genes, such as BRCA1 or BRCA2, can predispose someone to breast or ovarian cancers. Second, there are environmental carcinogens, substances like tobacco smoke, asbestos, or even UV radiation from the sun. Third, infectious agents like human papillomavirus, HPV, or Epstein-Barr virus, EBV, can trigger specific cancers. Finally, lifestyle factors such as diet, alcohol consumption, and obesity also play significant roles. We also categorize cancers by their tissue of origin. Carcinomas, the most common type, arise from epithelial cells, the cells lining organs like the skin, lungs, and gut. Sarcomas come from connective tissues like bone or muscle. Leukemias are cancers of blood-forming tissues, while lymphomas originate in the lymphatic system. So, to summarize the slide, cancer is essentially the failure of normal regulatory systems of a cell. When these systems break down due to genetic mutations and environmental triggers, we end up with cells that grow and divide when they shouldn't. And if these cells invade and spread, that's what we call cancer in the strict sense. This sets the stage for our next discussion. What are the common strategies that almost all cancers use to survive, grow, and spread? Hanahan and Weinberg, two researchers, synthesized decades of data into a powerful framework called the Hallmarks of cancer. This is what we'll dive into now. Back in 2000, two scientists, Douglas Hanahan and Robert Weinberg, proposed a groundbreaking idea. They argued that despite the vast diversity of cancers, from lung cancer to leukemia, there are a set of shared biological capabilities that all cancers must acquire to survive and thrive. They called these the hallmarks of cancer. This idea was later updated in 2011 to include new discoveries. Think of these hallmarks as the cheat codes that cancer cells use to break the normal rules of biology. If a normal cell is like a law-abiding citizen, cancer cells are criminals who exploit every loophole they can find. The 6 original hallmarks are: sustaining proliferative signaling, constantly telling themselves to grow; evading growth suppressors, ignoring signals that normally stop growth; resisting cell death, avoiding programmed suicide; enabling replicative immortality, dividing endlessly without aging; inducing angiogenesis, creating their own blood supply; activating invasion and metastasis, spreading to other tissues. Later, researchers identified 2 emerging hallmarks: deregulating cellular energetics, changing how they generate energy; avoiding immune destruction, hiding from or suppressing the immune system. And finally, there are 2 enabling characteristics that make all this possible: genomic instability, which provides a high mutation rate and raw material for evolution; tumor-promoting inflammation, where the body's own immune response ironically helps tumors grow. This framework has been incredibly influential because it allows us to understand cancer not as random chaos, but as a structured disease with predictable strategies. For you as students, It's important to grasp that almost every cancer, no matter the type, must achieve these hallmarks in order to become malignant. Let's start with the first one, sustaining proliferative signaling. Normally, cells require specific signals to grow and divide. These signals are like green traffic lights, and without them, cells remain quiescent, in a resting state. Growth factors, such as epidermal growth factor, EGF, bind to receptors on the cell surface, triggering signaling cascades inside the cell that eventually push it to enter the cell cycle. Cancer cells, however, are masters at hacking the system. They don't wait for external green lights. Instead, they create their own permanent green lights or disable the red lights entirely. There are three main strategies cancer cells use here. Autocrine signaling. The cell produces its own growth factors. It's like sending itself a constant divide now message. For example, some glioblastoma cells secrete PDGF, platelet-derived growth factor, which then binds to their own receptors. Overexpression of receptors. Some cancer cells produce far more growth factor receptors than normal. Breast cancers overexpress HER2, a receptor tyrosine kinase. This means even small amounts of growth factor can trigger massive, continuous growth signaling. Constitutively active signaling proteins. Mutations in downstream signaling molecules can lock them in the on position. The classic example is RAS, a small GTP binding protein. Normally, RAS cycles between active and inactive states. But a mutation can trap RAS in its active form, continuously sending growth signals to the nucleus, even in the absence of growth factors. From a clinical perspective, this hallmark is crucial because it gives us therapeutic targets. Drugs like imatinib block the BCR-ABL fusion protein in chronic myeloid leukemia, essentially cutting off the abnormal growth signal. Similarly, monoclonal antibodies like trastuzumab, Herceptin, target HER2 in breast cancer, reducing signaling intensity. So, sustaining proliferative signaling is essentially the cancer cell's ability to provide itself with never-ending instructions to grow. Without this, cancer cells wouldn't have the fuel to multiply uncontrollably. This is the first of many hallmarks, and it sets the tone for how cancer behaves as a disease. It bends normal biology to its own advantage. Now that we've seen how cancer cells sustain constant growth signals, let's talk about the second hallmark, their ability to ignore stop signals. In our bodies, cell division isn't just about having the accelerator pressed down, it's also about the brakes. Growth suppressor proteins act as those brakes, making sure cells don't divide when they shouldn't. Two of the most famous suppressors are RB1 and TP53. RB1, retinoblastoma protein. This protein controls the critical G1/S checkpoint of the cell cycle. It decides whether a cell has enough signals and resources to start DNA replication. If RB is intact, the cell cycle won't proceed until conditions are right. But if RB is mutated or inactivated, the checkpoint is lost. The cell keeps moving forward into division, regardless of whether it's safe. TP53, P53, often called the guardian of the genome. This protein detects DNA damage. If the damage is reparable, P53 halts the cell cycle until repairs are made. If the damage is too severe, P53 instructs the cell to undergo apoptosis, programmed death. But when TP53 is mutated, which happens in more than 50% of cancers, cells with DNA errors slip through, divide, and pass those errors onto daughter cells. Other tumor suppressors also play roles, like PTEN, which opposes PI3K/AKT growth signaling, and NF2, Merlin, which regulates contact inhibition. So how do cancer cells evade these suppressors? There are several strategies. Mutations that completely inactivate tumor suppressor proteins. Deletions of the entire gene. Epigenetic silencing, like methylation of promoter regions. A clinical example is retinoblastoma, a childhood cancer caused by the loss of both RB1 alleles. Another is Li-Fraumeni syndrome, where inherited TP53 mutations predispose individuals to multiple types of cancer. From a therapeutic perspective, restoring tumor suppressor activity is challenging. It's easier to block an oncogene than to replace a missing suppressor. But exciting research is exploring ways to reactivate mutant p53 or mimic its function. So, the second hallmark is all about cutting the brake lines. If the first hallmark is pushing the accelerator, the second ensures there's nothing to stop the car from crashing forward. Our third hallmark takes us into the realm of apoptosis, or programmed cell death. Normally, cells that accumulate too much damage or become unnecessary undergo apoptosis, a clean, controlled suicide that prevents harm to the body. But cancer cells are like rebels who refuse to die. They find ways to disable the death machinery, ensuring their survival even when conditions should kill them. How does apoptosis normally work? One major pathway is the intrinsic mitochondrial pathway, where damage signals activate proteins like BEX and PUMA. These proteins make pores in the mitochondrial membrane, releasing cytochrome C, which activates caspases, the enzymes that dismantle the cell. Another pathway is the extrinsic pathway, triggered when death receptors, like FOS or TNF receptor, bind their ligands, again activating caspases. Cancer cells resist cell death in multiple ways. Overexpressing anti-apoptotic proteins like BCL2. This is especially seen in certain leukemias and lymphomas. Loss of pro-apoptotic proteins, such as BX. Mutations in TP53, which normally triggers apoptosis in response to DNA damage. Defects in death receptors or downstream caspases. A striking clinical case is follicular lymphoma, where a chromosomal translocation places the BCL2 gene under control of a strong promoter, leading to constant BCL2 expression and resistance to apoptosis. Therapeutically, this hallmark has been targeted successfully. Drugs like venetoclax inhibit BCL2, reactivating the cell death pathway in certain leukemias. So, resisting cell death is crucial for cancer survival. Imagine a soldier that should fall when injured but refuses to die, continuing to fight in a way that harms the whole body. That's what cancer cells achieve here. Next, let's discuss cellular aging and how cancer cells cheat it. Normal cells are not immortal. Every time a cell divides, the ends of its chromosomes, called telomeres, get shorter. After about 50 to 70 divisions, the Hayflick limit, telomeres become critically short and the cell enters senescence or apoptosis. This is a natural safeguard against uncontrolled proliferation. But cancer cells bypass this limit by reactivating telomerase, an enzyme that elongates telomeres, essentially resetting the clock after each division. Telomerase is normally active in stem cells and germ cells, but is silenced in most adult somatic cells. In contrast, more than 90% of cancers reactivate telomerase, giving them the ability to divide indefinitely. A minority of cancers, about 10%, use an alternative method called alternative lengthening of telomeres, which involves recombination-based mechanisms. From a clinical perspective, telomerase activity is a potential diagnostic marker since its presence often indicates malignancy. Telomerase inhibitors are being investigated as therapeutic strategies, though challenges remain in selectively targeting cancer cells without harming normal stem cells. This hallmark, enabling replicative immortality, explains why cancers can continue to grow for years or even decades. While normal cells retire after their lifespan, cancer cells keep finding ways to stay young forever. Now, let's consider how tumors feed themselves. As a tumor grows beyond about 1 to 2 millimeters in size, simple diffusion of oxygen and nutrients is no longer sufficient. At this point, the tumor faces a crisis. Either it finds a way to supply itself with blood, or it will stop growing. Cancer solution? It hijacks the body's ability to build new blood vessels, a process called angiogenesis. Tumors secrete proangiogenic factors, the most famous being VEGF, vascular endothelial growth factor. VEGF binds to receptors on endothelial cells, stimulating them to sprout new blood vessels toward the tumor. At the same time, cancers suppress anti-angiogenic signals. This new vasculature is often abnormal, leaky, disorganized, and inefficient. But it's enough to provide oxygen, nutrients, and a pathway for metastasis. Clinically, angiogenesis is a key therapeutic target. Bevacizumab, a monoclonal antibody against VEGF, is used to starve tumors of their blood supply. Other drugs block VEGF receptors or signaling downstream. Interestingly, angiogenesis is also a double-edged sword. While it feeds tumors, the abnormal vessels can actually make drug delivery more difficult because of poor circulation. So, by inducing angiogenesis, cancers literally build their own lifelines, ensuring continued survival and growth. Perhaps the most feared aspect of cancer is metastasis, the ability to spread from the original site to distant organs. Primary tumors can often be removed surgically, but once cancer spreads, treatment becomes much more difficult. The metastatic process involves several steps. Local invasion. Cancer cells invade surrounding tissues by breaking down the extracellular matrix, ECM, using enzymes like matrix metalloproteinases, MMPs. Intravasation. They enter nearby blood vessels or lymphatic vessels. Survival in circulation. The bloodstream is a hostile environment. Cancer cells must avoid immune attack and survive physical stress. Some travel as clusters or with platelets to shield themselves. Extravasation. They exit the circulation into new tissues. Colonization. Finally, they must adapt to the new environment and grow. A key biological event here is the epithelial to mesenchymal transition, EMT, where cancer cells lose adhesion, eke adherent downregulation, and gain mobility. Metastasis is highly organ-specific. For example, prostate cancer often spreads to bone, while colon cancer frequently spreads to the liver. This pattern is partly explained by the seed and soil hypothesis. Cancer cells, the seeds, grow preferentially in tissues, the soil, that provide a favorable microenvironment. Clinically, metastasis is the main cause of cancer-related deaths. Understanding and preventing it is one of the biggest challenges in oncology. Finally, let's discuss the updates to the Hallmark framework. Emerging hallmarks. Deregulating cellular energetics. Normal cells primarily rely on oxidative phosphorylation in mitochondria. Cancer cells, however, often switch to glycolysis, even when oxygen is available, a phenomenon known as the Warburg effect. This allows them to generate energy quickly and produce building blocks for growth. Avoiding immune destruction. Our immune system constantly patrols for abnormal cells. Cancer cells develop mechanisms to hide from immune attack. They may express proteins like PD-L1, which binds to PD-1 on T cells and shuts down the immune response. Enabling characteristics. Genomic instability. Cancers often have high mutation rates due to defective DNA repair. This instability provides the diversity needed for cancer evolution and drug resistance. Tumor-promoting inflammation. Paradoxically, immune cells infiltrating tumors can release growth factors and proteases that help cancer progress. These updates remind us that cancer isn't static, it's an evolving disease. By studying these hallmarks, researchers can identify vulnerabilities that can be turned into therapeutic targets. So, to wrap up the hallmarks, cancer cells achieve immortality by systematically overriding the rules that govern normal cell behavior. Each hallmark gives them a survival advantage, and together, they create the malignant phenotype. Alright, class, we've now understood the hallmarks of cancer, the essential strategies that cancer cells use to survive, grow, and spread. But the obvious next question is, what is happening at the genetic level? What changes in DNA allow cells to acquire these hallmarks? The first category of cancer-related genes we need to discuss is oncogenes. Let's break down the word itself. Proto-oncogenes are normal cellular genes that play crucial roles in growth, division, and survival. You can think of them as the accelerators of the cell cycle. They push the cell forward when it's time to divide. When proto-oncogenes are mutated, overexpressed, or structurally altered, they become oncogenes, permanently pressed accelerators. The car, our cell, is constantly speeding forward, even when it should stop. How do proto-oncogenes become oncogenes? There are several mechanisms. Point mutations. A single base change in DNA can alter a protein's function. A classic example is the RAS gene. Normally, RAS cycles between on and off states, depending on signals. but a mutation can lock it in the on state, continuously sending growth signals downstream. Gene amplification. Sometimes, the cell produces too many copies of a gene. With too much protein being made, signaling goes out of control. For instance, amplification of MYC, a transcription factor, is common in several cancers, including Burkitt lymphoma. Chromosomal translocations. Pieces of chromosomes can break and reattach incorrectly. This can create fusion genes with abnormal function. A famous example is the Philadelphia chromosome in chronic myeloid leukemia, CML, where a translocation between chromosomes 9 and 22 fuses BCR and ABL genes, creating the BCR-ABL fusion protein, a constantly active tyrosine kinase. Overexpression of receptors. Sometimes the receptor for a growth factor is produced in abnormally high numbers, making the cell hypersensitive. An example is HER2/neu, a receptor tyrosine kinase amplified in certain breast cancers. Examples of key oncogenes. RAS family, HRAS, KRAS, and RAS mutated in 30% of all human cancers. MYC, a transcription factor that drives cell growth. HER2, in breast cancer, overexpression leads to aggressive tumor growth. BCR-ABL, a fusion oncogene, driving uncontrolled signaling in CML. Clinical significance. Oncogenes are typically dominant at the cellular level. This means that only one mutated copy of the gene is enough to cause problems. Compare this to tumor suppressor genes, which usually require both copies to be lost. This property makes oncogenes excellent drug targets. If we can block their hyperactive signaling, we can slow or stop cancer progression. A famous success story is imatinib Gleevec, which specifically inhibits BCR-ABL and has transformed CML from a fatal disease into a manageable condition. Similarly, trastuzumab, Herceptin, targets HER2 in breast cancers, improving survival dramatically. So, oncogenes are like accelerators stuck in the go position. They are the genetic drivers that keep pushing cancer cells forward, and understanding them has given us both diagnostic markers and therapeutic breakthroughs. Now, let's shift focus to tumor suppressor genes, the protective guardians of the cell. If oncogenes are accelerators, tumor suppressor genes are the brakes. They keep cell division in check, repair damage, and eliminate cells that could become dangerous. When these genes are lost or inactivated, the brakes fail, and cancer cells are free to divide without restraint. How do tumor suppressors work normally? They can act in multiple ways. Cell cycle regulators. Proteins like RB1 control critical checkpoints, especially the G1/S transition. Without them, cells enter DNA replication without proper checks. DNA damage sensors and repair. TP53 monitors DNA integrity. If damage is detected, it halts the cell cycle or initiates apoptosis. BRCA1 and BRCA2 repair double-strand DNA breaks. Apoptosis promoters. Some tumor suppressors directly trigger cell death when things go wrong. Key examples. TP53, called the guardian of the genome. Mutations in TP53 are found in more than 50% of human cancers. Without P53, cells with severe DNA damage keep dividing, accumulating further mutations. RB1, the first tumor suppressor discovered, associated with retinoblastoma, a rare childhood eye cancer. RB regulates the cell cycle's G1/S checkpoint. BRCA1/2. Mutations in these genes predispose individuals to breast and ovarian cancers due to impaired DNA repair. PTN, a phosphatase that opposes growth signaling through the PI3K/AKT pathway. Mechanisms of inactivation. Point mutations, common in TP53. Deletions. Entire gene regions may be lost. Epigenetic silencing. Promoter methylation can turn off tumor suppressor genes without altering DNA sequence. Inheritance and the two-hit hypothesis. Tumor suppressor genes typically follow the Knudsen two-hit hypothesis. This means both copies of the gene must be lost or inactivated for cancer to result. In sporadic cancers, both hits occur somatically. In inherited cancer syndromes, patients are born with one defective copy, the first hit, and need only one more hit for cancer to develop. For example, individuals with inherited BRCA1/2 mutations have a much higher lifetime risk of breast/ovarian cancer. Clinical significance. Tumor suppressors are more difficult to target therapeutically than oncogenes. It's easier to block an overactive protein, oncogene, than to restore a missing one. However, strategies are being developed to reactivate mutant p53 or exploit vulnerabilities created by tumor suppressor loss, a concept called synthetic lethality. For example, PRP inhibitors are used in BRCA-mutated cancers because these cells are already deficient in DNA repair pathways. So, to summarize, while oncogenes represent go signals gone haywire, tumor suppressor genes represent the loss of the stop signals. Cancer arises when both the accelerator is stuck down and the brakes are cut. Together, these genetic changes form the foundation for the hallmarks of cancer we've been discussing. When you hear the word chemotherapy, what comes to mind? Probably images of strong medicines, hair loss, and side effects. And you wouldn't be wrong. Chemotherapy has a reputation for being harsh, but it has also saved countless lives and remains a cornerstone of cancer treatment, especially for rapidly dividing tumors. What is chemotherapy? Chemotherapy refers to a group of drugs that kill cells or stop them from dividing. The key idea is that cancer cells divide faster than most normal cells, so drugs that interfere with cell division should preferentially affect cancer. Unfortunately, some normal tissues also divide rapidly, like bone marrow, hair follicles, and the lining of the gut, which explains many of the side effects. Mechanisms of action. Chemotherapy drugs fall into several classes, each targeting different parts of the cell cycle. Alkylating agents, e.g., cyclophosphamide, cisplatin, attach alkyl groups to DNA, causing crosslinks and strand breaks. Prevents replication and transcription. Antimetabolites, e.g., methotrexate, 5-fluorouracil, mimic normal building blocks of DNA or RNA. When incorporated, they block DNA synthesis or repair. Antimicrotubule agents, e.g., paclitaxel, vincristine, interfere with the mitotic spindle, halting metaphase. Topoisomerase inhibitors, e.g., doxorubicin, adipocyte, block enzymes that manage DNA supercoiling, leading to breaks during replication. Cytotoxic antibiotics, e.g., bleomycin, bind DNA and interfere with its function. Side effects. Because chemo targets dividing cells, normal tissues get caught in the crossfire. Bone marrow suppression, leading to anemia, infections, and bleeding tendencies. Hair loss, alopecia. Nausea and vomiting, due to effects on gut lining. Infertility, damage to gamete-forming cells. Secondary cancers. Paradoxically, DNA-damaging drugs can sometimes cause mutations in normal cells. Clinical use. Chemotherapy may be used alone, but more often it's combined with surgery, radiation, or targeted therapies. In leukemias and lymphomas, chemo can be curative. In solid tumors, it can shrink tumors pre-surgery, neoadjuvant, or prevent recurrence after surgery, adjuvant. Limitations. One of the biggest problems is drug resistance. Cancer cells mutate quickly and can evolve ways to pump drugs out, repair DNA damage, or bypass blocked pathways. This is why combinations of drugs are often used. Hitting multiple pathways at once reduces the chance of resistance. So, in summary, chemotherapy is like carpet bombing. It doesn't discriminate well between healthy and malignant rapidly dividing cells, but it can be life-saving. Modern oncology is gradually moving toward more precise therapies, but chemotherapy still plays a vital role. Now, let's move into one of the most exciting frontiers in modern cancer treatment, immunotherapy. The core idea is simple yet powerful. Harness the body's own immune system to fight cancer. Remember, our immune system is designed to recognize and eliminate abnormal cells, including cancer. But cancer evolves tricks to hide or disable immune responses. Immunotherapy is about reactivating those defenses. Strategies in immunotherapy. Immune checkpoint inhibitors. Normally, immune checkpoints like PD-1 and CTLA-4 act as brakes to prevent overactive immune responses. Cancer cells exploit these by expressing PD-L1, which binds PD-1 on T cells, shutting them down. Drugs like nivolumab, anti-PD-1, or atezolizumab, anti-PD-L1, release these brakes, letting T cells attack tumors again. This approach has revolutionized treatment of melanoma, lung cancer, kidney cancer, and more. CAR-T cell therapy. Here, doctors extract a patient's T cells, genetically engineer them to recognize a tumor-specific antigen, like CD19 in B-cell leukemias, and infuse them back. These modified T cells act as a personalized army against the cancer. Cancer vaccines. Unlike preventive vaccines, like HPV vaccine for cervical cancer, these therapeutic vaccines stimulate the immune system against existing cancers. Example: Sipuleucel T for prostate cancer. Cytokine therapies. Cytokines like interleukin-2 or interferons can boost immune cell activity. Advantages. Immunotherapy can produce long-lasting remissions even in advanced cancers. Unlike chemotherapy, which kills cells directly, immunotherapy trains the immune system, potentially giving durable protection. Challenges. Immune-related side effects. When the brakes are released, the immune system can attack healthy organs, causing colitis, dermatitis, thyroiditis, or pneumonitis. Not all patients respond. Some tumors lack strong immunogenic signals. High cost. CAR-T therapy and checkpoint inhibitors are extremely expensive. Now, students, let's shift gears and talk about targeted therapy, which really represents a modern revolution in cancer treatment. If chemotherapy is like dropping a bomb that kills every fast-dividing cell in sight, then targeted therapy is more like a sniper rifle. It doesn't aim everywhere. Instead, it focuses on one very specific weakness in the cancer cell. How do we even know what to target? Well, remember the oncogenes we discussed earlier? Those accelerators that get stuck in the on position? Or the overactive growth signals that refuse to shut down? Targeted therapy is designed to switch those off. Take chronic myeloid leukemia, CML, for example. For decades, it was a deadly disease. But once scientists discovered that CML is driven by the BCR-ABL fusion protein, they designed a drug called imatinib. This drug slips into the protein's active site and blocks its activity. Suddenly, those uncontrolled signals stop, and the cancer cells die off. Imatinib changed CML from a fatal illness into a chronic, manageable condition. That's the power of targeting a single molecular defect. Another great story is in breast cancer. Some aggressive breast cancers have far too much of a receptor called HER2. This receptor makes the cells hypersensitive to growth signals. Scientists developed an antibody, trastuzumab, that latches onto HER2 and blocks it. For patients whose cancers are HER2 positive, this treatment has dramatically improved survival. Now, of course, it's not all smooth sailing. Cancer cells are clever. Even if you block one pathway, they can sometimes find detours, mutating the target protein or activating an alternative survival route. That's why resistance to targeted drugs often develops. In practice, doctors may have to switch to second or third-generation drugs, or combine different targeted agents. Another exciting direction is exploiting what we call synthetic lethality. Here's a simple analogy. Imagine a bridge supported by two pillars. If one pillar is already broken, say, the cell has a BRCA mutation that weakens DNA repair, then if we deliberately knock out the second pillar, using a PRP inhibitor, the whole bridge collapses, but only in cancer cells. Normal cells, with both pillars intact, survive. This clever trick has led to successful treatments for BRCA-mutated ovarian and breast cancers. So, targeted therapy isn't just about killing cells, it's about understanding the biology, really digging into what makes a particular cancer tick, and then designing a therapy to exploit that exact weakness. It's precision medicine in action. Now, it's important to remember that while targeted therapies usually cause fewer side effects than chemotherapy, they're not completely free of them. Blocking EGFR, for instance, can cause skin rashes, and HER2 drugs can sometimes affect the heart. So, even precision treatments need careful monitoring. But overall, targeted therapy has shifted our mindset. Instead of asking, what organ is the cancer in? We now also ask, What mutations or molecular changes are driving this tumor? And increasingly, treatment decisions are based not just on where the cancer started, but on its molecular profile. This class is the future of oncology. We are moving from one-size-fits-all approaches toward deeply personalized medicine. And targeted therapy is leading that journey. All right, class, let's take a step back and look at the big picture of what we've learned today. Cancer is fundamentally a disease of uncontrolled cell growth, but it's much more than just cells multiplying wildly. At its core, cancer arises because the normal rules of biology, growth, repair, death, are systematically broken. These broken rules are captured beautifully in what we call the hallmarks of cancer. We talked about 6 classical hallmarks first. Cancer cells sustain proliferative signaling, essentially pressing the accelerator constantly. They evade growth suppressors, cutting the brakes that normally stop division. They resist cell death, refusing to undergo apoptosis, even when they're damaged. They achieve replicative immortality, bypassing the natural limit of cell divisions. They induce angiogenesis, building new blood vessels to feed themselves. And finally, they activate invasion and metastasis, spreading from the original site to distant organs, the feature that makes cancer so dangerous. We also covered emerging hallmarks, like altered metabolism and immune evasion, and enabling characteristics such as genomic instability and tumor-promoting inflammation. Together, these hallmarks explain how cancer behaves as a cunning, adaptive disease rather than just a collection of rogue cells. At the genetic level, we identified 2 main types of genes driving cancer: oncogenes and tumor suppressor genes. Oncogenes are like accelerators stuck in the on position. Think RAS, MYC, HER2, or BCR-ABL, pushing cells to divide uncontrollably. Tumor suppressors, on the other hand, are the brakes, proteins like TP53, RB1, and BRCA1/2, which are lost or disabled in cancer. The combination of a stuck accelerator and lost brakes is what gives rise to many of the hallmarks we discussed. Finally, we looked at therapeutic approaches. Chemotherapy is the traditional approach, attacking rapidly dividing cells, but with many side effects. Immunotherapy, the modern revolution, recruits the patient's own immune system to recognize and destroy cancer, using checkpoint inhibitors, CAR T cells, and vaccines. Targeted therapy is precision medicine at its best, zeroing in on specific genetic or molecular weaknesses in cancer cells, like BCR-ABL in CML or HER2 in breast cancer. The key takeaway is this: understanding cancer's biology, its hallmarks, genetic drivers, and vulnerabilities is what allows us to design effective treatments. The future of oncology is moving towards personalized medicine, where therapy is tailored not just to the type of cancer, but to its molecular fingerprint in each patient. So when you think about cancer now, don't just see it as uncontrolled growth. See it as a disease that cleverly bypasses nature's rules, adapts to its environment, and challenges us to understand it at a deeper level. And that understanding, from hallmarks to genes to therapies, is precisely what modern medicine uses to fight it.